So good afternoon. My name is Raquel Sánchez Valle. I'm a behavioral neurologist working at the Hospital Clínic of Barcelona. I'm currently the chair of the Dementia study group of the Spanish Neurological Society. It's for me a great honor to share this session on amyloid therapies with Dr. Estela Area-Gómez from the Centro de Investigaciones Biológicas CSIC de Madrid and Columbia University of New York City, and with Professor Bart Strooper from the Leuven Center for Brain Disease Research, Belgium, and University College of London, a UK-Dementia Research Institute in UK. I will just start with a brief introduction to the amyloid therapies. These are my disclosures. I must say that when I saw that the amyloid session was the session number 6, I thought that this was a challenge from the organization to the amyloid hypothesis, but let's wait until the end of the session to draw some conclusions. At this point, I will not perform a survey now, but I will ask you to choose which of these sentences do you think fit best to the history of amyloid. The first one is, I have not failed, I have just found 20,000 ways that don't work, from Thomas Edison. The second is success is walking from failure to failure with no loss of enthusiasm, from Winston Churchill. And the third is there are no shortcuts to any place worth going, from opera singers called Beverly Hills. You can think of this sentence during the talk. So we will start with the present. These are where are we now in the clinical? These are the regulatory agencies' approval for clinical use at the beginning. The FDA in the US with aducanumab in 2021, with accelerated approval, lecanemab in 2023, donanemab in 2024, in UK, lecanemab in 2024, and also donanemab some months later. But NICE decided against recommending anti-amyloids for routine use in the national health system due to its cost effectiveness, even if there is an appeal period ongoing. At the end, the EMA, lecanemab was approved in April 2025, and donanemab was recommended for approval in July 2025, even if we are still waiting for the resolution of the European Commission. But both in the UK and the European Union, the approval was exclude homozygous for ApoE4. We have had a marvelous session this morning of ApoE4, and maybe you can agree or you cannot agree with this decision of the EMA, but at the end is what we have in clinical use at this point. We cannot provide this neither lecanemab or donanemab to patients who are homozygous for ApoE4 based on safety. And now we are receiving, that's also present time, the first results with anti-amyloid antibodies in clinical practice. Anti-amyloid antibodies treatment for early AD is both feasible and safe in spare settings. There is still no data available on clinical efficacy, and we have the data from the results from the Washington University in St. Louis that only a minority of the patients receive at this unit receive anti-amyloid antibodies, about 6% of the total, about 16% of those with a CDR.5 or 1, and 22% developed ARIA, 6% developed symptomatic ARIA, 1% developed severe symptoms. With a new data that was not present in the clinical trials, patients with mild dementia had a higher risk of symptomatic ARIA than those with MCI. included in this treatment. And of course, this study underlined that we need have relevant needs for healthcare professionals and facilities to provide these patients the care they need for receiving these treatments. But this was a long history. Let's go to the past now. And this is a slide for the youngest people in the room. You know that in 1906, Alzheimer described the amyloid plaques. In 1984, the amyloid VIB-KU was identified. In 1987, the gene encoding amyloid precursor protein was described as a cause of familial Alzheimer's disease. In 1992, the amyloid cascade hypothesis was formulated, proposing that A-beta plaque deposition is the main pathogenic factor of AD, and that tau pathology, cell loss, and vascular damage are a direct result of amyloid deposition. In 1995, the prosalin genes were identified, causing familial Alzheimer's disease, and in 1999, the first positive results in mice with anti-amyloid therapies were published. It was 1999. This is the gene, the protein. This is the basic scheme of the metabolism of amyloid that my colleagues know much more than me, but just to summarize that there are two main metabolic pathways, the non-amyloidogenic and the amyloidogenic pathway. The disbalance between these two metabolic pathways produce the accumulation of beta amyloid in these very beautiful plaques, but very scary plaques in a patient. And the amyloid cascade hypothesis has a never-ending story of controversies. There are some of these key discussion points. As I'm a clinician, I will focus that the discrepancy between the amyloid deposits and the clinical symptoms has been argued as a problem for the cascade hypothesis. My view is on the contrary. I think that the fact that the tau pathology correlate better with neurodegeneration and clinical symptoms than amyloid pathology is not a problem for the hypothesis, but it's just a part of the hypothesis. But anyway, there are many pros and cons for this hypothesis. But I say that was it was in 1999 we received the first positive results in mice, and we have 20 years of negative clinical trials with patients. Many failures with both passive anti-amyloid immunotherapy and passive anti-amyloid neuroimmunotherapies with different drugs with different type of clinical trials. Then we received the first positive clinical trials with anti-amyloid immunotherapy, with antibodies, passive anti-immunotherapy. We will leave apart the aducanumab results, but we have positive for Clarity AD for lecanemab, a trial-based Alzheimer 2 for donanemab, with about 1,800 subjects each, and with a magnitude of effect, we will see later, of 27 to 36%, even if both populations at the design were not similar, so we cannot have a direct comparison between the both trials. Now we will explain the history of lecanemab. I think it's worth it. In 1999, Dr. Lanfer identified a mutation in the APP gene that causes autosomal dominant form of Alzheimer's disease in Sweden. Unlike other genetic forms of Alzheimer's disease, this family do not present the typical mature plaques that are observed in the amyloid PET. I mean that these families are amyloid PET negative. This observation led him to the hypothesis that soluble isophore, especially protofibrils, might represent the toxic form of amyloid, as these families without mature plaques or raw also develop symptoms of Alzheimer's disease. He founded the company BioRT and identified an antibody that binds to protofibrils. The problem, or not the problem, the serendipity was that it also binds to amyloid plaques. This antibody, lecanemab, was later co-developed with Eisai. I just also mentioned that, unfortunately, carriers of the active mutation were excluded from clinical trials, so we don't know if this drug works in these families. These are the primary outcome results, about 800 subjects, mean age 71 years old, 18 months, 1-to-1 lecam placebo, and patients included were in the mild cognitive impairment phase or mild dementia phase. All of them were amyloid positive by PET. The primary outcome was the clinical dementia rate in some of boxes, and after 18 months, there were a difference between those patients treated compared to placebo of 0.45 points in this scale. That was about 27% of clinical effect differences. Lecanemab also saw that it has a strong capacity to remove the amyloid plaques measured by amyloid PET, with and 66% of the patients were negative for amyloid PET at the end of the study. Donanemab, in contrast, it was focused in a peptide that was present only in stable, mature A-beta plaques. The study trial basis was also about 1,800 subjects, 18 months, mean age of 74 years old, also CDR 0.5-1, amyloid positive, and in this study, they stratify the patients or subdivide the patients based on the positivity in the Tau PET, in low, medium Tau PET participants, and in high PET participants. And they analyzed the results both in the low, medium Tau PET and in the total population. The results, the primary outcome was not the CDR, was a different clinical outcome, but it was observed also in the low, medium Tau population, a 35% of change, a different magnitude of change of the treated patients compared to placebo, and in the total population of 22%. This difference between the low, medium Tau PET and the total population was interpreted because the low, medium population was in an early stage of Alzheimer's disease, and probably the treatment, the donanemab, was more effective in this early phase of the disease compared to the total population, but really there was not a formal comparison between the low, medium Tau and the high Tau. Interestingly, both with lecanemab and donanemab, the clinical benefits increased with time, both within the trial, but more interestingly, beyond the trial, beyond the double-blind phase of the trial. So that's, but we still don't know how strong or how long is this increasing benefit with these drugs. Donanemab also could remove amyloid measured by amyloid PET. We can see here the difference, and almost 80% of the patients at the end of the study were negative for amyloid measured by amyloid PET. Interestingly, in this trial, the patients who became negative in amyloid PET were discontinued from the treatment. They stopped receiving the treatment because the design considered that they were amyloid negative. There was no reason to continue with the drug infusions. But in the study, we cannot see changes in Tau PET. And in both studies, both in the lecanemab study and in the donanemab studies, there were controversial results in marker of neurodegeneration. So we still don't know what happens really with neurodegeneration after these anti-amyloid drugs. And what about safety? The most frequent adverse event is infusion-related reactions. About every 4 or 1 every 3 patients have some infusion-related reactions that usually are mild and transitory, but the more scary and the more severe adverse events are related to the presence of ARIA. ARIA is amyloid-related imaging abnormalities that can be edema, effusion, or microhemorrhage. And it depends, I will not focus in the exact numbers because it depends on the trial. They have both drugs have new results with new data but that's a relevant adverse event that, as we saw in the Washington University results in clinical practice, are around 1 every 5 patients, and about 6% are symptomatic and 1% can be severe. Interestingly, ARIA-A especially is influenced by the APOE4 genotype, and that's why the EMA excluded APOE4 homozygous from the approval of these treatments in Europe and the UK. Putting together all these trials, both failures and success, we learned that the amyloid reduction results have an impact in clinical efficacy in the sense that the more the amyloid reduction, the more the clinical efficacy, and really to obtain relevant clinical effects, we need to clear almost up all the amyloid plaques we can measure with amyloid PET. I insist in the term what we can measure with amyloid PET because we can only measure the mature plaques with amyloid PET. We don't know what happens with the other species of the amyloid. And in that sense, we know that these trials that were positive with donanemab and lecanemab were targeting the amyloid plaque. It doesn't mean that the clinical effect is caused by the plaques, because probably removing the amyloid plaques are changing the equilibrium of the different species of amyloid. So we really cannot know at this point, or from my point of view, is which spice really is acting in clinics. And what about the future? First, I'm a clinician. Amyloid therapies is a challenge for the health system. That's something that the Spanish Neurological Association is working in, because we have to improve the diagnosis of Alzheimer's. We know that approximately 50% of the patients with mild cognitive impairment due to Alzheimer's disease are not diagnosed in Spain, so we have to have better tools to identify these patients. We will have to have quick pathways to refer these patients from the primary care to the memory units. And of course, we need in the memory units more tools, and of course, more health professionals with experience in these treatments. But I think that amyloid therapies in early Alzheimer's disease is a work in progress. We probably will see soon transition from intravenous to subcutaneous, because subcutaneous, it's more comfortable for patients, less expensive for the healthcare, and enhance patients' compliance. And also, we are already seeing new dosage titration schemes, changes in frequency and duration of these antibodies treatment based on improved efficacy and safety of new versions of current approved drugs, of new drugs. But we definitely need accurate, valid, and reliable data on efficacy, safety, and cost in the clinical settings and long-term clinical and biomarker data. And that's something that us as a clinician may have to do because probably the pharma companies will not provide this type of studies in the clinical setting with real life experience. But of course, the future is just to move from early Alzheimer's disease to preclinical Alzheimer's disease. We call secondary prevention study when we treat the patients when the amyloid deposits are already observed in the brain of the patients, and primary prevention when we cannot observe amyloid plaque deposition in the patients. At this point, there are thousands of participants in preclinical 80 trials in the world, so there will be some very interesting results in a few years, I thought. And I will mention now that this was everything about sporadic Alzheimer's disease, but we have also data on genetic Alzheimer's disease. This is a trial we participated in, and this study demonstrated that partial or short-term amyloid removal did not show significant clinical effects in familial Alzheimer's disease. However, long-term full amyloid removal potential delay symptom onset in a small set, only 22 patients, participants that were asymptomatic but carried the mutation, providing the first evidence of potential ED symptoms prevention in this population. And now we are starting the primary prevention trial in this population with genetic Alzheimer's disease with participants who does not have amyloid plaques in their brain. For me, the summary about amyloid therapies is that the contribution of amyloid therapies to the AD field is yet to be determined. And a question probably for the discussion, if we have time, is how do we imagine the future? And if I start with several sentences, I finish with another sentence that the best way to predict the future is to create it. So we are here to create the future to our patients and people at risk of Alzheimer's disease. So thank you very much. This was an introduction to the issue, but the real talks start now. First, with Estela, Dr. Estela Area-Gómez, the role of MAM in the regulation of APP processing. Thank you, Estela, for being here.
Okay. Okay, well, first of all, of course, thank you to the Fundación 100 and the organization for this very nice invitation to the amyloid session, where I'm not going to talk about amyloid. And I'm pretty sure that I'm just the opening act for the start of the day, which is Dr. Strooper. It's an honor to be here with him. And I'm going to talk about not amyloid, but a very close related relative of amyloid. Forget about the title right now. Sadly, I don't have anything to disclose. But I'm going to tell you a brief story about how we just came up with a potential idea of what can be going on in Alzheimer's disease. Well, one disclosure, I'm going to talk about lipids, but very briefly. So this is the team that started in 2004 and in New York with all these beautiful people, and now it continues in Spain with only girls. I don't know why. But so I don't need to tell you that one of the main achievements of the eukaryotic cell in evolution is compartmentalization. We tend to forget about it. So in each compartment in the cell, okay, there is different pathways confined where there is a specific environment that allow these different pathways to be very finely regulated. I would also to remind you that these compartments are membrane compartments. So each specific organelle has a membrane with a unique composition acquired during differentiation, during evolution, and any change, this kind of like a specific lipidome allows every organelle to talk to their environment and regulate every single pathway that we have. But one question remains, because sometimes, pathways do not occur in only one organelle, but in two. It's kind of like they have to come together to complete the pathway. So is it true that, as you know, cholesterol can be synthesized in the ER, and it can be used to synthesize bile acids, oxysterol or cholesterol esters. Also, palmitate, a fatty acid, can be used to synthesize different phospholipids, or it can travel to the peroxisome where it's going to be oxidized and then transferred to the mitochondria to produce ATP. So how does this work? Okay, so imagine that this actually works through contact sites between organelles. So for instance, a fatty acid in the cell can be activated in the endoplasmic reticulum to form fatty acid coenzyme A, which is the form that the cell understands that it can use that fatty acid. If the peroxisome, this fatty acid in the endoplasmic reticulum can result into the synthesis of different phospholipids. But if the peroxisome is bound to the endoplasmic reticulum, that fatty acid can be rerouted to form other types of lipids, such as plasmalogen, bile acids, etc. On the other hand, also, if peroxisome, instead of being bound to the endoplasmic reticulum, it's bound to the mitochondria, as I mentioned before, that fatty acid now is going to be oxidized in the peroxisome and then transferred to mitochondria to be further oxidized to produce ATP. So in here, what I'm saying with that is that contact sites are not new compartments, are just places, different areas where the different organelles can get in touch and share metabolites. Those are temporal, very transient binding, like a kiss and run kind of type of contact that allows also the spatial regulation of different pathways. So in many instances, when the contact sites are dysfunctional, those pathways cannot be properly regulated. In the lab, we have a favorite one, of course, which is the interaction between the mitochondria and the endoplasmic reticulum. So when mitochondria binds to the endoplasmic reticulum, it binds ER through a specific domain. This domain is called mitochondria-associated ER membranes, or MEM. MEM is not, again, a new compartment. It's a transient domain that is formed in the endoplasmic reticulum, and then it binds to mitochondria to crosstalk, to maintain the crosstalk between both organelles. So, as I mentioned before, these contact sites are temporary. So this is kind of like a summary. So there is a formation of a domain, and then mitochondria rise to this domain, and then what happens is that a lot of different enzymes get clustered in these domains, and to regulate different functions included glucose, bioenergetic, phospholipids, cholesterol esters, fatty acid, metabolism, sphingolipids, cholesterol, and lately also autophagy. So it is a functional domain, meaning it is an ER domain that carries and clusters a lot of different functions. So in the lab, in order to understand the regulation of these contact sites and the formation of the MEM in the ER, what we do is we assay different enzymes that are localized in this domain and activated when it is formed, such as, for instance, and this is a lot of lipids, I understand, such as the esterification of cholesterol. We know that cholesterol in excess arrives to the endoplasmic reticulum, and the cell can get rid of that cholesterol by esterifying it and confining those, or packaging those, into lipid droplets that are going to be secreted by the cell. Also, another function that occurs in the MEM and in the crosstalk to mitochondria is the synthesis of phosphatidylserine and its conversion to phosphatidylethanol. And also, sphingolipid metabolism, it's very well known that its regulation is centered at the MEM and crosstalk also to mitochondria and to Golgi. So, but one question remains, so how is MEM formed? So I would like to emphasize one thing, which is MEM is a lipid raft. Okay, what does it mean by a lipid raft? A lipid raft is a domain that is formed spontaneously with no ATP necessary just due to increases in the local concentration of cholesterol. So what happens, and it creates this kind of like a floating islet or raft in the middle of the membrane. The membrane is like a sea of oil. And this lipid raft will be like a piece of butter that is floating there, a solid, almost solid, rigid domains. So how it happens is that cholesterol, it is always in membranes and it's highly hydrophobic. So when there is an increase in cholesterol in a membrane, it's never homogeneously distributed. So what it does, it actually binds together and it surrounds itself by other lipids to avoid water. So, and basically hiding inside the membrane. So cholesterol moves passively to get clustered together. Along with cholesterol, a lot of different cholesterol-binding proteins move passively, also without no need of ATP, and can get clustered together in this raft or this membrane. So cholesterol-binding domains then, due to this change in the lipid environment, can change their conformation. And not only that, now they are dragged together and they can interact with each other. This happens in one very well-known localization for all the audience, which is in synapses. Synapses are planar. And they're planar because they are full of cholesterol. And sometimes, when we say dimerization of receptors in the synapses, what we are saying actually is that two different receptors come together because cholesterol, they are cholesterol-binding, cholesterol-avid proteins that they are forced to be kept together using cholesterol as glue. So cholesterol is continuously moving around the cell, and it actually follows a very clear distribution. So once cholesterol is enriched in these domains, the cell can actually get rid of cholesterol and dissolve this temporal lipid raft by different enzymes that are going to be in charge of eliminating cholesterol from the membrane, such as cholesterol esterases, oxidases, etc. So this is a staining of cholesterol in one fibroblast in blue. And as you can see, there is a very clear gradient of cholesterol in the cell, because as soon as cholesterol is increased in some of the membrane, it's going to trigger a lot of different signaling mechanisms. So that's why we can consider, or in the lab we consider cholesterol distribution as a signaling mechanism. So that's the MEM, actually. The MEM is a lipid raft. So we wonder whether, there is some sort of like a crosstalk between cholesterol regulation and the MEM domains in the ER. As you well know, cholesterol can be synthesized in the cell, okay, by a very big pathway, the mevalonate and posisqualin pathway that occurs in the endoplasmic reticulum. However, most of the cells in our organism do not synthesize cholesterol. They uptake it from the diet in the form of lipoproteins. So how it works is that the lipoprotein is going to be internalized, and the cholesterol inside the lipoprotein is going to be used first to replenish the cholesterol in the plasma membrane. Then the excess of cholesterol, it's going to manipulate, or it's going to deform the plasma membrane and form this endosome, a cholesterol-rich endosome. This cholesterol-rich endosome is going to bind to a lysosome, and it's going to form an endolysosome that is going now to be delivered to the endoplasmic reticulum. As soon as it arrives to the endoplasmic reticulum, by the pathway of Brandt-Golsing, is going to inhibit the activity of cholesterol biosynthesis enzymes as well as its expression. So we wondered what is the relationship between MEM and cholesterol. So we did a very naive experiment. So we took a lot of different cell types and exposed them to cholesterol. And then as control, we use also one enzyme that is called sphingomyelinase. It's a very old-fashioned enzyme that mobilizes cholesterol from the plasma membrane to the ER. And also to prove that indeed, whether it will be the role of cholesterol synthesis, we also use different statins, atorvastatin, lovastatin, and simvastatin. And we measure MEM activity through phospholipid synthesis and cholesterol esterification. Long story short, statins didn't do anything. However, as you can see here, increasing concentrations of cholesterol over time increase the activity of the MEM and its crosstalk with mitochondria through the synthesis of phosphatidylserine and phosphatidylethanolamine. Also, we measure cholesterol esterification, and as you can see, the increases in cholesterol activate cholesterol esterification in the MEM by one enzyme called acetyl-CoA acetyltransferase, or ACAD1. And also, as a result, we have an increase in the formation of lipid droplets in the cell. Also, we were able to replicate that by the use of sphingomyelinase. That, as I mentioned before, sphingomyelinase is an enzyme present in bacteria that mobilizes cholesterol from the plasma membrane, and it forces its delivery to the endoplasmic reticulum. So what we found in here, this is a staining of cells with philippine that stains free cholesterol in blue. And I don't know if you can appreciate it, by the incubation with the sphingomyelinase induces the appearance of a lot of droplets formed with free cholesterol, an increase in the uptake over wild-type levels or untreated levels, and a very significant increase in the formation of cholesterol esters. Likewise, we actually found that these increases, the incubation with sphingomyelinase or incubation with cholesterol also increases the physical apposition between mitochondria and endoplasmic reticulum. And of course, also the activity, the phospholipid transfer between ER and mitochondria. So our first conclusions or deduction is that MEM is a temporal domain that is formed by the internalization of cholesterol from the plasma membrane and is delivered to the endoplasmic reticulum. In here, in the endoplasmic reticulum, when MEM is formed, there is a clusterization or an aggregation of different proteins in here that now can interact or change their conformation. So that's why we define MEM as a signaling domain, as a functional domain that regulates many different functions, as I mentioned before: lipid metabolism, calcium, autophagy, mitochondrial dynamics, and every day there is a new function. And of course, it's going to regulate cholesterol esterification and the formation of lipid droplets. I just want to underline one concept. This is what happens with a protein. It is in a normal membrane, let's put it this way, and this is a protein that changes its conformation when cholesterol arrives to the endoplasmic reticulum. And not only that, these proteins, which are cholesterol-binding proteins, allow for cholesterol to be stabilized. So there is a crosstalk between the cholesterol and the cholesterol-binding protein, both forming this raft functional domain. So you will be wondering, so where is this woman in an Alzheimer's meeting? And as I mentioned before, MEM regulates many different functions, including gamma secretase activity. So as you well know, gamma secretase is in charge of the cleavage of the C-terminal fragments of APP, C83 and C99. And of course, in healthy cells, we find that most of the cleavage, or most cells, most of the cleavage occurs through the non-amyloidogenic pathway, and this cleavage occurs in non-raft domains. However, in patients with Alzheimer's disease, what we have is an increase in this cleavage, in the amyloidogenic cleavage of APP. And as I mentioned before, I mean, a long time ago, we found that presently it's enriched in these MEMS. And I'm glad to report that many other groups have been able to replicate this. So how does it work? So you have one field of research that is cholesterol, and they all agree that cholesterol travels from the plasma membrane towards endoplasmic reticulum. And then you go and review the literature on APP cleavage, especially the amyloidogenic cleavage of APP, and they overlap. Basically, it's the same, but of course, I mean, depending on what you study. So whenever you put them together, I mean, you found that APP has a high affinity for cholesterol, and it can be internalized together with cholesterol in these cholesterol-rich endosomes that fuse with a lysosome to form an endolysosome, and now base, which, as you well know, I mean, the pH of the optimal pH is acidic 6. So, and now it can get delivered, this cholesterol together with C99, arrives to the endoplasmic reticulum and allows, due to its capacity, the capacity of C99 to bind cholesterol, it helps cluster all this cholesterol in the MEM domain, and also different proteins that are now regulating autophagy, calcium, mitochondria, lipids, etc. And as I mentioned before, gamma secretase also gets dragged towards the C99, and as soon as the lipid raft is formed, is cleaved into amyloid beta and AICD. This is an unhealthy situation. What happens if all of a sudden, I mean, you expose cells to very high increases in cholesterol, exogenous cholesterol, what we know that it actually triggers the MEM, the formation of the MEM. So what you get actually is this, a very high increase in cholesterol internalization and the cleavage, an increase in the cleavage of a base, and an accumulation of C99 into the endoplasmic reticulum. So these are some pieces of data that we have in the lab in different cells that actually, I mean, has been shown already before by Catherine Marker, how, I mean, the increase in membrane cholesterol in neurons recapitulates, not only, I mean, you get an increase in amyloid, also you get an increase in C99 and an increase in A-beta 42 over A-beta 40, as well as the hyperphosphorylation of tau. So what happens now in a familial AD? Okay, so in familial AD, we know there are mutations in gamma secretase in presenting 1 or 2, as well as mutations in APP. For a long time, I mean, due to the increasing amounts of amyloid that you can see in patients with mutations in gamma secretase, we thought that mutations in gamma secretase were a gain of function. Nevertheless, I mean, whenever you actually describe whether an enzymatic activity is increased or decreased, you cannot look at the substrate, at the product only. The activity of enzymatic activity is defined by the ratio between the product, the substrate, and the product. And what we have found is that, of course, there is an increase in amyloid in Alzheimer's disease, but there's even a higher increase of C99, and this C99, what we observe, is not homogeneously distributed in the cell, but it accumulates in these areas of the endoplasmic reticulum in close opposition to mitochondria or the MEM. This is, I mean, just a Western blot showing that indeed we have a high increase or significant increase of C99 in familial Alzheimer's cells and also in sporadic Alzheimer's cells. So C99, what we found is that C99 increases the formation of the MEM and triggers the internalization of cholesterol in sporadic and in familial Alzheimer's cells. As you can see, I mean, we found that cholesterol uptake is highly upregulated in familial and sporadic AD. MEM activity is highly upregulated too at the expense of cholesterol biosynthesis de novo. So this is our first deduction here, and this is one idea that we have in the lab. We are probably wrong, but what we believe is that familial Alzheimer's mutations result is a loss of function that increases or accumulates C99. SAD risk factors that increase cholesterol uptake is going to actually converge into the increases in C99 at the MEM. So there is some sort of like crosstalk between these two features in the disease. And that could actually explain many things that we have been observed in AD, which is APP function and cleavage regulated by lipids, by cholesterol, of course, by a lipid raft. So metabolism of cholesterol is going to depend on APP, and APP cleavage is going to depend on cholesterol. And not only that, what we found is that C99 contains a cholesterol binding domain that acts as a prion, meaning as soon as you drop C99 in a membrane, without no need of enzymatic activity, no nothing, just by its cholesterol binding domain, it changes completely the structure of the membrane and activates, moves and distributes different enzymes and can activate them. No need of enzymatic activity. Just for those that you believe that lipids are boring and don't do anything. They do a lot of stuff. So, okay, so what are the consequences of this? Okay, as I mentioned before, MEM regulates a lot of different functions and specifically a lot of different lipid enzymes. Such as, sphingolipids, phospholipids, fatty acids, cholesterol esters, and lipids are very interesting because they regulate the permeability of membrane, the function of the membrane. They also regulate protein conformation depending on the different lipids and the saturation of the membrane. And not only that, they can also regulate curvature, as Jorge mentioned this morning. So, we did we are studying several different lipid proteins that localize and are regulated at demand, but one of which we found by proteomics, and it's well known, which is SACL4. A different SACLs. What is ACL? It's acyl-CoA synthesis. What they do is that all fatty acids that we have in the cell, in order for the cell to use them as food or as structural blocks, they need to be activated into fatty acyl-CoA. And SACL, that can be used for beta oxidation or can be used for lipid synthesis. One specific SACL has been in fashion lately, which is ACL4, by its relationship to ferroptosis. Why? Because SACL4 prefers arachidonic acid. So what it does, it binds to arachidonic acid and activates it. Now arachidonic acid can be included into phospholipids, or it can be used also towards the synthesis of different inflammatory reactions, as well as cannabinoid neurotransmitters. So arachidonic acid is kind of important. So what we found in here is that when we measure the activity, the activation of arachidonic acid by SACL4 in different subcellular fractions of different tissues and cells, we find that this is actually a validation of previous results, that it is enriched in the MEM compared to other compartments. But not only that, in wild-type cells, compared to just VIB-KU, when we expose them to a sphingomyelinase, that I remind you, it just forces the entry of cholesterol and pushes the formation of the MEM, there is a significant increase. This increase can be also phenocopied by the overexpression or accumulation of C99. And this overactivation we can block by impairing the traffic of cholesterol with this inhibitor. We were able to replicate that also in wild-type cells exposed to inhibitors of gamma secretase, which is going to increase the levels of C99, and we can rescue them by inhibit base with a base inhibitor in here. Also, we validated that in APP double knockout cells, where compared to wild type and the empty vector, as you can see, C99 can rescue a little bit, the activity of SACL4. So using these cells, we tracked what is the fate of arachidonic acid once it's being activated in these cells that are overexpressed in C99. And we found that this fatty acid is incorporated mostly into phospholipids. And specifically, although it is incorporated in several phospholipids, majorly it's incorporated into phosphatidylcholine when C99 it's accumulated and overexpressed. And this we can actually detect in lipidomic cells, in the lipidomics of specific cells that express C99 wild type, and this is a C99 form that is mutated in the cholesterol binding domain, or even in wild type cells when we expose to inhibitors of the gamma secretase and accumulate a C99. And as you can see, we observe here, this is a heat map. In color, we can see all the changes that are statistically significant. And what we found is that an increase, significant increase in phosphatidylcholine species that are bound to polyunsaturated fatty acids and majorly to arachidonic acid that we can recapitulate here when a gamma secretase is inhibited. And indeed, compared to wild type, we can see that cells that accumulate C99, there is a significant increase in polyunsaturation. That's what in membrane unsaturation. So what does it have to do with AD? So what we decided, I'm sure that you're very aware of that there is a very clear increase in lipid polyunsaturation in Alzheimer's cells. That has been going on in the field for a long time. And so we decided to test the SACL4 activity in Alzheimer's cells, familial Alzheimer's cells, different fibroblasts and different tissues, as well as in sporadic AD. And we do find an increase in the activity of SACL4 over control. And also, we find the resultant increase in phospholipids bound to arachidonic acid or to polyunsaturated fatty acid that we can rescue when we silence SACL4 in familial AD and also in sporadic AD. Again, as before, this results in an increase in phospholipid polyunsaturation in Alzheimer's cells. So this is our second and last deduction here. So we believe that there is a crosstalk between cholesterol, APP, and the MEM, and that there is some contribution in this trio in Alzheimer's disease, because as soon as you increase cholesterol uptake, you form the MEM and you activate gamma secretase. However, if you increase gamma secretase or C99, if you decrease gamma secretase and accumulate C99, you observe an increase in the MEM and an increase in cholesterol uptake. So these three pathways are co-regulated and are deficient in Alzheimer's. Once MEM is altered in the disease or in the context of these cells, we're going to start accumulating a lot of different lipid alterations on the course and the progression of the disease that is going to result in membrane defects, especially damaging to those cells whose membranes are very, very active, such as neurons and microglia. Also, we can explain with that why Alzheimer's cells present quite early alterations in calcium transference, given that MEM actually is rich in this kind of enzymes that co-regulate the transfer of calcium between ER and mitochondria. And also, mitochondrial failure, which is well known that occurs in the disease. We believe that is a symptom or a consequence of these alterations in the MEM. So why is this important? How can we translate that into something for the clinics? I'm not a clinician, but I collaborate with some of them, and especially with two of my beloved collaborators, Gloria Patricia Cardona and the late Francisco Lopera, that as you well know, they've been collecting patients from this cohort of familial AD with mutations in presenilin 1 or the PISA mutation. And I'm going to introduce an idea that includes one concept that is a little bit weird in the field, time. So We've been collecting samples, serum samples, and different tissues from this cohort between the ages, the ages between 8 and 12 carriers of this mutation with different ApoE4 isoforms, 13 to 20 years old, 20 to 30, and 30 to 40. It is well known, by CIMOA, that around 25-year-old, you can detect already phospho-tau 217, and amyloid in blood is quite high, in CSF is quite high. And you start seeing the positions of plaques around the 30th, which in this decade, in the last decade, you can also detect, PET problems, and cognitive decline. However, and this is very preliminary result, I hope that in the next meeting I can show you, very clear data. What we have found is that at 8-year-olds, we can already see lipid alterations due to defects in MAM regulation. So what do we believe is that I'm not anti-amyloid, I'm not anti-plaques. I think that it's a little bit more complex than that. I think that it's a matter of time. So at the beginning of the disease, we think that there is an accumulation of C99 and a high production of amyloid. However, the ratio C99 amyloid is higher. So C99 is going to start a lot of different detrimental metabolic dysfunctions in the cell. Over time, not only those alterations, but the constant depositions of plaques are going to accumulate in the disease, meaning we believe that this can be the driver, or an early driver at least, that can start a cascade of events that is going to end up in what we call Alzheimer's disease. And with that, I'd like to introduce the MAM lab, as well as the Inflamam lab. This is Jorge Montesinos, recently independent postdoc, that is going to studying all these theories and the role of C99 cholesterol in microglia regulation. And of course, I would like to thank all my collaborators and to you, and please take care of your man. Thank you so much.
We'll leave the questions for the end of the session. And now is the turn of Professor Bart Strooper, who will present a talk entitled 5 Inflection Points in Alzheimer's Disease, from Amyloid-Driven Inflammation to Tau-Mediated Neurodegeneration. Thank you for coming, and the floor is yours.
Thank you. So thank you for the invitation. I apologize, but my time is completely consumed, so I will try to be brief, and I will skip some slides so that we end in a reasonable time. So what I would like to do is to give you a different view on Alzheimer's disease, which tries to integrate all the different informations we have. Because I think I think that, well, I'm getting here the same problem with the, what? This one, yeah, okay, thank you. So I think, first of all, it's important to define precisely Alzheimer's disease. We have now the tendency in the field to say that everything is Alzheimer's disease. All kinds of neurodegenerations are Alzheimer's disease. I don't think that's the way to solve this problem. We need to be precise, and I think that the neuropathology is defining Alzheimer's disease. So we talk only about tau pathology as an Alzheimer's disease feature in the context of amyloid plaques. And second, I think next to amyloid plaques and neurofibrillary tangles, the granulovacular neurodegeneration is an important aspect of this disease. And so any hypothesis should include these 3 different neuropathological events. I also think that we are driven too much by the symptomatology of Alzheimer's disease, defining it as a dementia. And we know, and I will just briefly repeat that, we know that Alzheimer's disease is in fact starting many years before there is any clinical symptom. And I think that it's like in cancer. You don't define cancer with cachexie, you define it as a tumor which is present, even if it has no clinical symptoms. And I think with Alzheimer's disease, we should start to think in the same way. If there are amyloid plaques or neuronal tangles, it's not normal, even if there are no clinical symptoms. Sorry. So how do you study this disease? We don't have many clues towards what causes Alzheimer's disease. There is a lot of speculation. But again, I think that the only real clue we have is the genetics. And Alzheimer's is mainly a genetic disorder, much more than Parkinson's disease. And that's why I show you this scheme, which has been shown already, which summarizes what we know about the genetics of Alzheimer's disease. And so this graph is complicated, but what you see in the x-axis is the abundance or the frequency of the SNPs in the population, so from very rare to, I don't know whether this pointer works, from very rare to the left to very abundant in the population. And on the y-axis, you see how penetrant these mutations are, how much is your chance to get Alzheimer's disease. And so then you see two groups here. So you see the familial Alzheimer's disease where the mutations are causal. If you have that mutation, you get Alzheimer's disease. And then you see these other genes which increase your risk. So they are not necessary and not sufficient to get Alzheimer's disease, but they will enhance your chance to get Alzheimer's disease. And so I think that you can only study these genes in the context of already initiated Alzheimer's disease. So they are probably an answer on amyloid plaque accumulation. I will explain you that further. So let's first have a look to the familial Alzheimer's disease. You all know that there are mutations in amyloid precursor protein and in the presenilin genes, and that they affect APP processing. Despite what the geneticists say, that when they find the gene that the problem is solved, for instance, with presenilin, it takes many, many years to understand what really happens. And so for presenilin, it was indicated in the previous talk that that was the clue to the amyloid hypothesis. It's true, but it was not the finding of the gene, because if you read that paper, presenilin was associated with tau, with calcium, etc., etc. And so it's only when we started to really do the molecular biology of this protein that we started to understand how presenilin mutations cause Alzheimer's disease. I don't have the time to go through all the literature here, but just in a summary, so for the mutations for amyloid precursor protein, it's very simple. They are clustered around the A-beta sequence, and they increase the production of A-beta peptide. And they usually, almost all of them cause an increased aggregation of the amyloid peptide. And so that's why we think that aggregation of the amyloid peptide is the initiator of Alzheimer's disease. So then we have the presenilin mutations. As you see, this blue subunit is presenilin. It's part of the gamma secretase, which was discussed already. And the gamma secretase cleaves amyloid precursor protein in the membrane. You can see that there on the drawing. It cleaves in the membrane, but it has an interesting capacity. It cleaves a first time, and then it gives consecutive cleavages. And so the shorter the A-beta peptide becomes, the less stable the interaction is, and at a certain moment, this A-beta peptide will be released. And so the clinical mutations in presenilin destabilize this blue subunit, so it becomes less stable. All the mutations in presenilin are amino acid substitutions. And so So I had to film here. This blue subunit is very labile in the gamma secretase complex. And so when you have a mutation, this becomes even more unstable. And so what you have is that this consecutive cleavage, that the interaction between the enzyme and the substrate, so between the gamma secretase and the APP, becomes destabilized. And so you get faster release of longer A-beta peptides. And so the more destabilizing the mutation is, the more of these longer A-beta peptides you produce. And there is even a correlation between this destabilization of the gamma secretase and the age of onset of the familial Alzheimer disease cases. So this is really showing that aggregation of A-beta, abnormal production of aggregation prone of A-beta is sufficient to trigger the whole amyloid and Alzheimer cascade. So this is also the basis of the amyloid hypothesis as it was formulated 20 or 30 years ago. The problem with the amyloid hypothesis is that it has never explained Alzheimer's disease. So it's an arrow hypothesis. So the amyloid, you see here again, the amyloid plaques, the tau pathology, and the granulovacular degeneration. And if you read the amyloid hypothesis, there is a link of the amyloid pathology to the tangles and to the neuronal loss or the synaptic failure. but it has never been really shown and demonstrated how these links are made. And I think that has really strongly misled the whole field for certainly the last years. Because if you don't understand how amyloid induces the rest of the pathology, you also don't know what you have to expect if you remove amyloid from a patient. And so I think that the model I will present here will start to help to understand this a little bit better. So the first question is, what does amyloid, how does amyloid plaque, is that damaging or not? And there are patients their brain is full with amyloid and they don't have dementia, so this has been used against amyloid hypothesis. There are the animals, which I think basically show that it's not enough to accumulate amyloid to get neurodegeneration. So I think it's fair to say that amyloid plaques can be at best a trigger of the disease, but that they are not a driver of the pathology. And so there are additional factors which will contribute to the neurodegeneration. And so the first thing we asked is, if you have amyloid plaques in a mouse brain, even if it doesn't induce tau, does it do anything to other proteins? And I mentioned already the sporadic Alzheimer genes, which indicate that neuroinflammation might be important. So the question is basically, is there anything which is similar to neuroinflammation? Bad word, by the way, it's astrogliosis and microgliosis. So, but is that, I use neuroinflammation because everybody's using it, but I'm not happy with it. Let's be clear about that. But so, this neuroinflammation, is there any signs of neuroinflammation around the amyloid plaques? And that's the experiment which we did a couple of years ago. We did spatial transcriptomics and we looked at the transcripts, the changes in transcripts around the amyloid plaques. I'm sorry about it, a bit tired. So what you basically hear is a circus plot. These are all genes which are changed around the amyloid plaques, and on the right hand, you see the amyloid plaque, and on the left hand, you see the circus plot, which indicates how different genes change their expression and are correlated with each other in changing their expression. And so these were genes. In green are astroglia genes, in blue are microglia genes, and in orange are made also astro and microglia genes, but when there is no amyloid plaque, you see that these genes, the blue and the green genes, are somewhat correlated in expression, and these orange genes are basically not expressed or not changing. And now you see what happens when we increase the plaques. So you see that here, and you see that the circus plot becomes denser and denser, and you see that there is correlation between the astrocyte and the microglia genes, and you see also that many of these orange genes start to be involved in this PIC network. We call it plaque-induced genes. And so you see here, for instance, ApoE gets involved, TREM2 gets involved, complement gets involved, a lot of other interesting genes get involved. So here you see the gene ontology. So what we see is that around these plaques, even in a mouse model, you get an induction of a neuroinflammation, which is characterized by classical complement cascade, lysosomal dysfunction, antigen processing and presentation, something very important, as you will see later, oxidation, reduction processes, immune responses. So there is indeed, even in the mouse, a very interesting neuroinflammation which is induced by the amyloid plaques. So we get rid of all this discussion that amyloid plaques is just a bin in which you put some amyloid, etc. No, amyloid plaque is a beta-sheeted structure, and the microglia do not like beta-sheeted structures. They think it's a bacteria, as somebody mentioned already today. So now is the problem, of course, how do we investigate this in human microglia? Because if we look to the response of microglia, the DAM response has been very nicely demonstrated. But if you look to the cloud, the single-cell cloud of the mouse, microglia responding to amyloid plaques, it's very simplistic. It's a complete different cloud than what you get below, microglia isolated from brain, human brain. And then you can discuss, is this a human-specific thing? Or is it because in the human brain, this is not a response to amyloid plaques, but it's in the late stage, it's the neurodegeneration, etc., etc. All these discussions in the literature. For me, the question is, what is amyloid plaques doing on human microglia? And so we need, I think science exists in reduction. You need to reduce the problem to specific question, and then you can build further, again, to complexity. So we decided that we needed absolutely to study human microglia in this context. Because if you look to the risk genes for Alzheimer's disease, and you look which genes are expressed in mouse microglia, you see that at the left. But you see that if you take human microglia, that at least 30% of genes relevant for Alzheimer's disease are not expressed in the mouse, but are expressed in human. And if you want to understand genetic interactions, which is important, then you will lose very rapidly track if you use a mouse model to understand how the inflammation goes. And so that's why we developed this, this model where we transplant human microglia in a mouse brain. So we need the mouse brain to get the pure amyloid plaque context, and then we need, of course, the human microglia in the brain to get a human microglia response. And so you see here an example. This works wonderfully. It works very well, very reproducible. It works with any iPSC cell that we have tested. And if you use the fire knockout, as you see here, that's a mouse in which genetically no microglia are produced. You can have 100% human microglia chimera. You see that at the right side. So every white dot is a human microglia. And on the left side, you see the mouse microglia. So you see they nicely sit in the place where they should be. And so then we did a lot of things with this microglia. You can read that. So one of the things which I'm not going to discuss here, but you can read the paper from Mancuso et al., where we looked to the cell states of this human microglia in the presence of amyloid plaques. And it's much more complicated than the mouse response. We get a kind of DAM response, but mainly an HLA response, an antigen presentation response in the context of amyloid plaques. And we get also cytokine responses. But I'm not going to explain that today. I'm just going to show you data which are not published yet, but which I think are extremely exciting and showing the strength of this approach. Because as I said, the important thing for sporadic Alzheimer's disease is to understand how SNPs, different genetic combinations, increase your risk for Alzheimer's disease. And since we think it's in the microglia that they have their effect, we need to have models to allow to study that. And so what we did is we took 31 different iPSC cells from different patients with an increasing risk for Alzheimer's disease, and we decided to put them in villages. I mean, we differentiate them together and we transplant them all together in a mouse so that the environment is the same. So what is different is the genetic makeup of the different iPSC cells, but the environment of amyloid plaques, the mouse brain, is the same. And so we can then compare how the genetic variability in the iPSC cells affect the microglia response. And so here this is briefly explained. So we have several villages. We transplant them in an APP, a human, which is a non-amyloid plaque forming control mouse, or we transplant it in the NLGF mouse, which is an amyloid plaque forming mouse quite rapidly. And so we can transplant, we can get the microglia in it, and then we can take them out, and we can do single-cell RNA sequencing. And because every person has his own unique genetic code, we can use that to know what cell that we isolated comes from which patient. So we can, with bioinformatics, we can find that out. And so here you see the cloud. We had more than 200,000 cells and a lot of data. And again, I'm going to summarize it just first of all. Here you see again this cloud, which I just showed you, but now the stains indicate where the cells from the different patients are located. And you see that there are very different responses. So every BR or whatever L is code for a different patient. And you see that in this cloud, they occupy different spaces. So that means that the transcriptome of this microglia is different. And since the only difference they have is the genetic makeup, this tells you that your genetic makeup very much determines how your microglia respond to amyloid plaques. And then we needed to make a choice. What kind of response are we going to investigate here? Because we cannot do multiple testing with such a small sample. So we decided to look for many reasons, which I don't have time. But as I said already, this HLA response, this antigen-presenting response, is a very important response of the human microglia to amyloid plaques. It's a very advanced stage where the microglia, which are normally homeostatic and supporting cells, become antigen-presenting cells. So that's a very different phenotype than what they usually have. And so what you see here on the right is every every patient, and you see this red diamond there, that's the mean HLA response of all the different cells. And you see it's really increasing when you do it in amyloid plaque. The black line is the HLA levels that we get in the control condition. So you see a real induction, but then you see for every patient that it's different. So there are patients which have a very strong response, and then there is even one patient which has a lower than normal HLA response. So this shows you very clearly that HLA response, antigen presentation, is influenced by our genetic risk. And so then the really interesting thing comes here, where we determined the polygenic risk score for each patient, and then looked how that translated in this HLA response. And lo and behold, I mean, this is really, we didn't expect that it would be so clear, but you have a linear correlation, a very good correlation of 0.7 with a high statistical certainty, that there is a direct correlation between your genetic risk of Alzheimer's disease and this HLA response, so this inflammatory response to amyloid plaques. So I come back to a scheme. I promised you that I wanted to explain a little bit differently how I think that Alzheimer's disease can be studied. And so what I basically would like to do is to define critical inflection points in the disease. We all agree that there is a progressive process there where you start with amyloid plaques, but amyloid plaques are not sufficient. You need different steps to come to the final clinical phase, which is brain atrophy and dementia. And so I explained to you already that a beta accumulation is the only trigger you need basically to get this whole cascade going. We know that from the FAD patients. And then I showed you that these amyloid plaques induce a relevant microglia, and I think the astroglia should be involved as well, because the microglia talk with the astroglia, that they create a neuroinflammatory environment. But now the big question is, how do you go from there to tau pathology? And so that's really the major question in the Alzheimer field. You cannot say it's tau or it's A-beta. I mean, they are integral part, and we know that the tau pathology is different in Alzheimer's disease from the tau pathology you get when you put a mutation in tau, which is the modeling we use for tau pathology in Alzheimer's. In my opinion, it's not very relevant for Alzheimer's disease because the P217 biomarker is not present in FTD. And second, if you look to the cryo-EM structures, it's very different in Alzheimer's disease versus the FTD patients. So you cannot just use FTD as a model for Alzheimer's disease. What we need is tau pathology induced by amyloid plaques or by the neuroinflammation, as you can see here in this suggestive scheme. So we thought maybe the story about humanized models is probably also true for the neurons. And so what we did is we use again, we say what happens with amyloid plaques. The mouse neurons don't develop tau pathology, so maybe if you transplant human neurons into the mouse, we might see that pathology. And it's really, it was really exciting to see that. So what you see here on the top are human neurons, they are green labeled, in a mouse brain. 18 months after transplantation, you can really keep them very long. And you see they are quite nice. And then you see below the same neurons, iPSC, these are embryonic stem cells. You see the same embryonic stem cells. They are differentiated to neuronal precursors, and then we transplant them 4 days after birth in brains of RAG2 immune-deficient mice, and then they grow. We just leave them there, and we look at different stages, what happens with these neurons. It's very difficult. I tell it very simple, but there is a huge variation in this transplantation. So the microglia transplantation works 95%. The human neurons varies between 20 sometimes and 80%. So it's really not easy. It takes a lot of time to get to this data, but they are here. So what you see in the lower panel is the amyloid plaques, the blue, and then what's really remarkable is the red stainings. It's 88, PHF1, and MC1. So 3 markers for tau pathology. MC1 is a pathological one, which are not seen in the mouse neurons, which are just laying at the site. So conclusion, is this tau pathology? Well, we did then some isolation of the tau pathology, sarcosylin soluble, and you can see here in that panel with the green arrows, the gold, the immunogold, which is immunogold labeling tau antibodies, and you see these are nice fibrils. And then the most interesting thing is here, so we took plasma from these mice, and we looked for human biomarkers, because these are human cells, they have tau, so we expected that the phospho-tau biomarkers would be there. And you can see very clearly that in the amyloid transplanted, we get phospho-tau 181, and we get phospho-tau 231 upregulated, so telling you that the pathology we see in these human neurons is likely relevant for Alzheimer's disease. And so we have neuronal cell loss. Now I'm going to go a little bit faster. But so it's for decades that people are discussing how are neurons dying. And I hear somewhere today that that's not important anymore. I honestly disagree, because I think that the loss of neurons is really the step towards irreversible dementia. Even if synapse dysfunction, etc., is important, I think that real problem in the fifth inclination point is neuronal cell loss. And so here, Sriram did stainings for several cell death pathways, and here in red, what you see is these vesicular structures, and this is phospholipid kinase 1 on the right side, and phospho-MLKL at the lower panel. And these are markers for the necroptosis pathway. And necroptosis is a very specific regulated cell death pathway. The phospho-MLKL goes to the membrane, make a hole there, and the cell dies. And it's typically linked to inflammation. So we thought this is very interesting, right? So we did a lot of work. The first thing I did was talking to Dietmar Thal, a real neuropathologist who knows what Alzheimer's disease is, and asked, did you ever see necroptosis in patients? And he did the stainings, and here you see these beautiful stainings here with phospholipid K1, phospholipid K3, phospho-MLKL. It are the big neurons in the hippocampus which develop this granovascular neurodegeneration. So we knew that CK1 delta stains this granovascular neurodegeneration. But you see that also this guys which cause necroptosis do the same. And so we can inhibit the loss of the neurons by using necroptosis inhibitors. I'm not going into the details, but this is a point I really want to make. So here you see another staining of human brain with CK1 delta, which is the classical biomarker for granovascular neurodegeneration in Alzheimer's disease. And you see that it colocalizes with phospho-MLKL, which is the executor of necroptosis. But what I think is that this is a protective mechanism. Because neurons, when they are dead and they disappear. If the membrane is lysed, you will not see them. And we see a progressive accumulation of GVDs and of these necroptosis markers in patients. So what I think is that the neurons die from necroptosis, but that those which can put these necroptotic executors into a vesicle, they can live a little bit longer. And so I just made here a scheme, very complicated, but don't worry. So on the right side, you see the triggers for necroptosis. You see there among them TLR4. And so then you form this square here at the right side, which is the necrosome. And then the necrosome sends the phospho-MLKL to the membrane and gives lysis. And there are two protective mechanisms known from cancer research. One is that it goes to multivesicular bodies and gets in vesicles, or at the cell surface, this necroptosis tunnel can be incorporated in an endocytic vesicle and can be set aside. And so that's why I think that the GVDs we see in Alzheimer's disease indicate neurons which are buying some time before they will be killed. So I'm going to leave the MEC3. So this is the most highly expressed transcript. It's a non-coding RNA, and it's involved in the neuronal cell death, but I'm going to skip that for reasons of time. And so the last point I want to make here is how important is phospho-tau in this pathway? Because that's the eternal discussion. Tau is the executor and amyloid is, I don't know what, if you don't like the amyloid hypothesis, and if you like the amyloid hypothesis, amyloid is doing everything and it induces tau pathology. I think it's much more interesting to think in the way I try to explain this here, which is the cellular phase of Alzheimer's disease, where you link the different steps by changes in cellular responses and where you try to find mechanisms. And so I asked George Scheres and Sofia Lofstam whether they could give me some tau seeds, which were characterized by their cryo-EM. and they make them synthetically, so there is no problem of inflammatory cytokines, etc., which you have when you isolate tau seeds from a human brain. And so we use that to induce in human neuronal cell culture in vitro, derived from our PSS, PHF tau, and you see that here indicated in red. And the question is that sufficient to induce necroptosis? And you see that in green, the phospho-MLKL vesicles in these neurons. So showing that indeed it is the phospho-tau. Once you get to this phospho-tau step, you get in this fourth inclination point. And so here is the summary. So the first inclination point is the formation of amyloid. In familial Alzheimer's disease, it's of course the mutations, but you can think about inflections, or you can think about high temperature, fever, or you can think about other things which destabilize this gamma secretase APP interaction, and which give you more A-beta, and which might then accumulate. Then you have this, the second inclination point, which is this neuroinflammation, which I explained to you. I put microglia also in this biochemical A-beta phase, because I don't have time to explain that to you, but if you delete microglia from the brain, you get less amyloid plaques. And I think it's an absolute mistake to always say that microglia are clearing amyloid. They are with much too little folk to do that, actually, but they take amyloid in, they condense it, and they secrete it as amyloid fibrils. But that's another story. So 1 is amyloid plaque formation, 2 is the neuroinflammation, 3 is the tau induction, and that can be either directly by A-beta, but I doubt that A-beta has a direct effect on the neurons. But I think it's the inflammatory context which make that the neurons react to the cytokines, etc., and go into hibernation. So it's not good that these neurons are very active when there is a neuroinflammation happening. So what the neurons do is say, oh, my supporting cells are clearing some misery here. I will not do any hyperactivity. And so I go in hibernation. So it's a maladaptive response to that inflammatory context. And it's very clear that there is a lot of experiments to be done here to understand what mediates that step. And then we come in the neuronal tau phase. So I show to you, once you have phospho-tau, we know that it seeds and that it can be transmitted and induce further tau in other places. But I showed you also that it's an important element in cell death. And then we come in the fifth inclination point. I hope it's here. Yeah, voila. So, and that's the one where everybody's talking about. When you do a clinical trial and you don't show anything on the fifth inclination point, then you will have no drug. I mean, that means that we would never have cholesterol medication, because if you think about what it means in that context, cholesterol medication giving after the patient had a heart infarct. And so that's what we do with the amyloid drugs. We want to see a miracle after the cell death and the tau pathology is already present. So, and that's the reason why these drugs don't work very well, in my opinion. And there is some indication in the data that we discussed that if you treat early, before the phospho-tau is there, that you have a much bigger effect. But anyhow, that's another discussion. So this is the summary then. Primary, secondary prevention, as we discussed, will be on amyloid, just like with cholesterol. I think it's a mistake to put all our money on antibodies. I showed you already. Yeah, I showed that in 5 minutes. I'm very sorry about that, that it became so late. The antibody is a very expensive thing to do prevention. It's never going to work. It's too costly, too much side effects, too difficult to do it. And I'm really disappointed that the knowledge we have around gamma secretase, which I explained to you, if you stabilize that gamma secretase with a gamma secretase stabilizer, then you can drive all the A-beta to shorter A-beta peptides, which remain soluble. And we have a lot of evidence that it works. And Roche has actually a phase 2 clinical trial showing that this drug is safe in patients. And so I expect that if we ever do primary prevention, it will be with a gamma secretase stabilizer. But that's also another story. Okay, so very last thing, antibodies. I mean, a lot of talk and a lot of speculation. Lecanemab recognizes soluble oligomers. Donanemab recognizes a pyroglutamate on a plaque. But they have exactly the same thing. There are a little bit differences, but in principle, they clear completely amyloid, and they have all ARIA side effects, and the clinical benefit in the cohort where it was tested is not spectacular. So I wanted to understand what these antibodies did. So we have amyloid plaques, and lecanemab binds to this amyloid peptide, or amyloid plaques, I think. Well, we will show that it binds amyloid plaques. And then on the other side, you have another part on the protein, which is the Fc fragment. And an Fc fragment interacts with microglia. So the first question I had with lecanemab, despite that it's considered as kind of zinc, which binds oligomers, etc., I don't believe in it because quantitatively it doesn't make any sense. You need some catalytic cell biological mechanism to have it cleaning the amyloid plaques. So human microglia, so we did an experiment. Thanks to our transplantation experiment, you can just use lecanemab. You don't need to make a murine version, etc. And so what you see here basically is that lecanemab doesn't clear the amyloid plaques if there are no microglia. So that shows you that you need microglia to have this effect. And so if you look to lecanemab, it's an antibody, and the Fc fragment has the LLP, 3 amino acids which are important for antibody phagocytosis, antibody-dependent cellular toxicity, and complement binding. So we mutated the LALA-PG, the L in an A, the L in an A, and a P in a G. And we used that. And here, this is a very nice summary of all the data. So what you see, the upper panel is what we have when we treat mouse with microglia with lecanemab. You see that there is almost no amyloid plaque in the third column here. The purple color indicates the antibody, and the green color indicates a beta stain. So we clear amyloid plaques when the microglia are there. But if you use lecanemab with these 3 amino acid substitutions, what you see is that you don't clear amyloid plaques, and you see huge binding of lecanemab actually on the amyloid plaques. So I think that's a working mechanism. So the antibody binds the amyloid plaques, gives the Fc fragment to the microglia, and the microglia change their style, because that's a real question here. Why do microglia not clean amyloid plaques? They are clustering there. And why do they do it when there is an antibody? So there must be something changing in the microglia. And I think that's very important because we are talking about beneficial and malignant responses of the microglia. So this must be a beneficial pathway. So we did single cell sequencing, and we found that lecanemab acts via the induction of specific genes related to phagocytosis, interferon metabolism, antigen presentation, and unfolded protein response. It's actually a very limited program. It sounds a lot, but there are only 150 genes which are different between lecanemab-treated microglia and not treated microglia. And so one of the major changes, you see it at the complete right side, is SPP1 or osteopontin. It comes from a lot of experiments, but here also in our experiment, we see that lecanemab induces this strongly. And so we decided to check it. And so we added lecanemab, we took an amyloid plaque, we took a slice from a mouse with amyloid plaques, and we put microglia on top of it. If you don't give antibody, they don't clear the plaques. If you give an antibody, they clear the plaques. But now we didn't give antibody, but we give SPP1 of osteopontin. It's very confusing. So SPP1 is the gene name, and osteopontin, OPN, is the protein name. So we give no OPN, or we give 1,350 nanogram ml OPN. In the presence or absence of microglia, you can see already that we can clear the amyloid plaques when we give this high dose of OPN. And on the right side is the real quantification, and it's very important. The blue is in the absence of microglia, and the red is in the presence of microglia. And see that it's sufficient to add osteopontin, which is induced by lecanemab, to this experiment to clear the amyloid plaques. So showing you that there is indeed a mechanism. So this is the end. I explained to you all the 5 dots here. So primary and secondary prevention we discussed. Of course, there will be in the inflammation some interesting aspects. Maybe we could just make something which activates the Fc receptor. So we get rid of all the antibody stuff, we cross-link the Fc receptor, and we do something, or we induce osteopontin, or we do other things, because this is just the beginning of the story. There are good things to be done there in the inflammation. I agree with what was discussed about REM2. We can also go into that if you want. But the problem is that the inflammation, I put it here in the second box, but there is also some inflammation. There is some microglia involved in the beta generation. And there is also for sure some effect of microglia on the tau pathology. And so I think one of the big questions, apart from understanding the mechanism in the tree here, what inflammatory or what protein, what receptor induces in the neurons this hibernation reaction. That's for me the major question. And the second major question is what aspect of the neuroinflammation are driving and what aspects of the neuroinflammation are protecting. And I think we have a fantastic biomarker, especially phospho-tau217, to study this phase 3 also in patients. And so then for phase 5, where we are all working for therapies, for me there is only one possibility there, and that's give some neurons back to these patients. And if you would have said that to me 3 or 4 years ago, I would have said you are completely crazy. But now that we can do these transplantations and also looking a little bit to Parkinson, we could maybe consider in a couple of years that we can restore a memory. But before you do that, and that's the mistake with the Parkinson, you need first to stop the pathological process. You cannot put a healthy cell in a disease context and then hoping that its healthy neurons are not going to be sick also. So these are my acknowledgements. I thank you for your time and sorry that it's so late.
So thank you very much for this exciting talk. Now the session is open to questions. First, Pascual, how much time do you have? We finish. We can take some questions. Pascual, please.
Wow, beautiful talks, really amazed. Thank you very much. I'm a neurologist and I am also involved in genetic epidemiology. And a frustration for us is polygenic risk score. How can we use this in the clinic? I mean, you have shown us that it could be relevant, that it give us a context. And do you think the polygenic risk score from the GWAS, or maybe we can learn more from the expression analysis, could be helpful some way in the clinic to the patient?
I mean, I was surprised that we saw this link. So since then, I'm a bit more positive about it, because I think there's a lot of hand-waving with PRS. And so the whole assumption there is that it's a linear accumulation of the different risks. And then if you think about risk, it's not anything meaningful. It just means that this SNP is more present in the group which you define as patients versus the control. And so if it's more there, then if it's 100, if it's 55 against 50, then you say your risk is 5%, etc. So I mean, if you start to think mechanistically about it, I think it's much too early, and it's typical for the genetics. I think that they think that if you have the gene and the gene risk, that you have the solution. While I think that the gene and the gene risk is the start for mechanisms. But to answer briefly, I think that there will be a time that we understand it's different, because this polygenic risk score is an accumulation of all the things. And so we were happy now to dissect a little piece out of it for the microglia. I think it's luck, but it shows the concept. And so if you do more iPSC cells and do other assays, for instance, differentiating between neurons and see whether there are neurons from certain patients which develop faster tau or faster necroptosis, then we will start to dissect, and then we can make meaningful clusters of genes, and then I think it will be really informative.
Alberto?
Well, thank you for the excellent talks. I have one question. Recently, it was described that gamma secretase has hundreds of substrates, and the mutations in presenilin, the pathology or the disease looks quite similar to a sporadic AD. Isn't this surprising that this is not lethal, or what happens with all the hundreds of substrates are also affected, and why is the disease in the end so similar?
I think we understand that now. But the interest in gamma secretase has become very low. But we understand now basically the whole thing. So all the mutations in presenilin which cause Alzheimer's disease are substitution mutations. That means that it's not really loss of function mutation, it's also not really a gain of function mutation. So the mechanism is basically, as I explained, that it makes the complex a little bit less stable and that you disturb the enzyme substrate interactions. And so the first cleavage happens with the whole protein, so where you have maximal interaction. So the signaling function of gamma secretase is the less affected by the mutations. And so you have a few mutations where you have a little bit of effect there, but most mutations don't affect the signaling part of the gamma secretase. And so it's basically once you have cleaved off the intracellular domain, that this consecutive cleavage happens, and then, of course, the shorter it becomes, the less stable your interaction becomes. And so the mutations mainly affect that. And so Lucia Chavez-Gutierrez has published a fantastic paper in molecular neurodegeneration where she showed that each mutation has a different age of onset, and each mutation destabilizes this gamma secretase to a larger extent. So the more it destabilizes, the earlier your age of onset. And it correlates with the length of the A-beta peptide. So for instance, the more severe mutations give you A-beta 43. So everybody thinks A-beta 42, but that's all correlation. So some mutations cause A-beta 43, and which give you a very fast aggregation of amyloid plaques. So that's one. And so on the other side, the signaling, first the explanation is there are a few where the mutation is so severe that if you have homozygotes, that you will have signaling dysfunction, but these people will not be born, basically, because you will affect the notch signaling pathway. So most people are heterozygote or have relatively mild, whatever you call it, relatively mild mutations. And so, so in the heterozygote situation, the wild-type allele is perfectly capable to compensate for any loss of function. We know that if you make a plus-minus mouse, president plus-minus mouse, these mice are perfectly normal. So the effect of the mutations on the other substrate is minimal. And that's the reason. And so just to make this to the end, because it's very important for the gamma secretase stabilizers. So the whole negative feeling about gamma secretase inhibition comes from gamma secretase inhibitors. Semagassestat was an inhibitor. And so if you use an inhibitor, then you block all the signaling of all these different substrates. And of course, you get side effects. I put a paper about learning by failing because I didn't understand why the field didn't take that to learn from it instead of saying we run away from that for in my opinion, the best drug target. But the stabilizers, they do the reverse of the mutations. So what you do is you bind to an allosteric site in the gamma secretase complex. And so what you do is you stabilize the substrate enzyme interaction. And so what you get, and we did the experiment, what you get is shorter A-beta peptides. So you can, even in a presenting mutation, you can go from 43 and 42 to more 38 and 37 peptides. So that's why I think, and that's why I'm looking very much forward to the Roche trial, which is a bit under the horizon because all the fuss is now about their shuttle antibody, etc. But I think in the long term, we need cheap anti-amyloid drug, which you can take on your 45 if you are at risk, and which you take like cholesterol medication to get your A-beta peptide balance right. And for all the other parts of the disease, once you are in the disease cascade, you will need additional therapy because at a certain moment, well, I explained, this neuroinflammation will become chronic. And even if you take away amyloid, it will still continue. And once you have tau, we know that from FTD, once you have tau, you can take amyloid away. It has done already its work. And so you will need these additional drugs.
So, I don't agree with the enzymatic
No, I
thing.
thought so, but...
you know me. But yeah, plaques and amyloid is really bad. So I really hope the antibodies work, but I don't think it's going to solve the primary problem.
I have a question for you. In your last slide, you recommend us to take care of mom. How do we take care of mom?
I don't know. That's always the question, how to treat mom. There is a whole move now, I'm very happy about it, actually, that they're focusing on mom therapies. I don't know. I'm not a clinician, and every time that I think about intervention, I mean, my brains get crazy. I am inside the cell. I don't even get outside the cell. I trust Alberto will do something.
Okay.
So, the Colombians.
So Pascual is asking us to finish the session. So, thank you very much, Estela and Bart. Thank you very much to the audience.
So I'm going to call the 2 winners to come on the stage. We need to be quick, so please, if you can come, we will give you your award. So our 2 winners are Alba Lucard Sánchez and Minerva Martínez Castillo. Please come to the stage.
¿Ella sola o todos?
Todos, hombre, yo también. Ah, vale. Well, now I think whoever wants to join the city tour, we are going to meet outside the building. So yeah, thank you very much.